Hey everyone, I just want to say thanks to the organizers, Juniper, Laurent, and Josh. You all have done an amazing job putting on a conference in what are certainly trying times. I am nothing but impressed with how well organized everything has been so far. And I just want to thank you sincerely for inviting me to give a keynote here. This is definitely one of my favorite communities that I'm involved in, and a place that I find a very natural home for my own work. And so, with that, I'll just jump right in. So, my title is New Frontiers in Alife: What Was Old is New Again. What I don't mean by my title is this idea that there's nothing new under the sun. That's not at all what I'm trying to say about Alife. And in fact, I'm hopefully going to convince you that I'm trying to say exactly the opposite. But before I get into what exactly my title does mean, I wanted to tell you a little bit of history about myself. So, I went to a small liberal arts college called Xavier University. It's in Cincinnati. People from the States probably know it mostly because they have a relatively good basketball team. They're pretty commonly make it relatively far into the NCAA championship, but it's not a huge college by any means. There were about 4,000 students when I graduated. And they had a computer science department, and that was what I majored in. And this is all of the people that were in one of the required courses, is artificial intelligence this year. There were so few students that basically any classes that were taught by one of the CS professors were required. And in this class, we got to play with these, we got to build and program these robots. And this is the first time I was really exposed to evolutionary computation. So some of the things that we did on the robots included things like subsumption architecture, but also programming the robots to evolve their controllers using sort of input that we gave it, but still using evolution as a way to solve problems with computers. And this was kind of fascinating to me. It was so much fun. I even managed to dig up this old video of our robots kind of going head to head with one that someone thought was the best robot and would clearly beat ours. One of the strategies towards the end of this project was to try and find the light before any other robots did. And one solution was to just push all the other robots off, which is what our solution was going for. And it didn't end up pushing off this robot in the challenge because it just never actually found the light, which, that was a short-sightedness on our part. But you can see we built a robot that was able to push other robots, including this robot that did win the tournament, by just sheer force off the table. So we're quite proud of that robot. After majoring in computer science at Xavier University, I did an internship with AT&T Research Labs. So this is one of the campuses of what used to be Bell Labs. And this one in particular was named after Claude Shannon, of course, the inventor of information theory. And here I was mostly working on social networks. So my research wasn't so oriented towards evolution at the time. I knew that evolutionary computation was something I was interested in. That's what really drove me to Michigan State to work with the big group of people there that were doing evolutionary computation at the time. But while I was in New Jersey and New York City, I spent a lot of time going to the Strand bookstore. And this is an amazing bookstore, and just browsing books at libraries and bookstores is one of the things that I've found myself missing a little bit more than I expected during all of this pandemic lockdown. But while I was there, I found this book, Evolution. And I didn't know who Carl Zimmer was at the time. I didn't really know all that much biology. I actually avoided taking biology classes in college because I was so scarred by it during high school that I just avoided it entirely. And I felt like maybe I should learn more about this thing that I was going to spend potentially the next 5 or 6 years of my life working on, at least from a computational standpoint. But I'd say this book certainly changed the trajectory of my career. It really opened my eyes to the idea that evolution was a problem that we were still working on solving. I thought up until this point, evolution was sort of an accepted fact and there weren't open questions behind it. that we knew everything we needed to know. At least that's what it seemed like to me as a computer scientist that was implementing evolution in these evolutionary algorithms. His new book, Life's Edge, which is not out yet, looks to be really, really fascinating, I think, especially for our community. So as a plug, don't forget to catch Carl Zimmer's keynote tomorrow in the Symbiosis and Artificial Life workshop. It's from 1435 to 1455, should be a short but fun one. So I ended up at Michigan State, and I was fortunate enough to join this group of PIs called the Devo Lab. And this isn't everyone that was part of the lab. It's just all the people that showed up for this particular picture day. If anyone who's done a picture day knows. And as soon as I got there, it was sort of clear that there wasn't a singular focus of the group. It was really interdisciplinary and really, really split up between kind of what I would call more traditional engineering problems and more traditional biological problems. And so sort of to represent this, I've got people as either robot heads or squid heads, and Charles is there, and kind of a combo of the two. And so my job my first time in this lab was to figure out sort of what direction I wanted to go. And I told you about reading the book on evolution, and that really opened my eyes. But at the time, I was still a computer scientist. I'd still been working on robots. I still really enjoyed robots. So my first project was working on evolving controllers for these little robots. And I found some videos of a couple of strategies that evolved while I was working on this problem. And this is kind of the classic evolutionary computation story where we reward something like how far a little walking table gets from where you start it. And evolution comes up with some pretty interesting strategies, ones that we maybe wouldn't have come up with ourselves. So things like this table on the left that really wants to be a biped, or this one on the right that just kind of cartwheels through the world. And this was cool, but it was kind of surprising that really I wasn't seeing tables that sort of wowed me more than a couple of rare instances, right? And biology doesn't have this problem. Biology is full of kind of amazing creatures and just surprises. And so I became really interested in why we weren't seeing these surprises in our evolutionary computation systems. And so I feel like one quote from Darwin that is often, it's overquoted, but I really appreciate it because I think it lays out a hypothesis for why the world is so complex, why nature looks so amazingly complicated. And he says that it's because these constructed forms, so different, are dependent upon each other, right? And so it's this interaction between organisms that seems really important. And that led me down kind of the path of being interested in understanding how organisms influence each other's evolution. And in particular, I became most interested in antagonistic coevolution. So in this case, there's this tension between populations where one's pushing and the other's pulling, and they're constantly kind of going back and forth. And I became particularly fascinated by the kind of work people were doing with bacteria and phage. And phage are viruses that infect bacteria. And I just found that people were really asking fundamental questions about the way evolution worked. And they were doing this experimentally in the lab with real biological organisms that were evolving, and people were able to study this in real time because bacteria and phage evolve so quickly. And this totally blew my mind. I had no idea people were doing this, and I just became completely enthralled with this idea. And I wanted to ask those same sort of questions. I wanted to use these computational systems that I was studying to ask these questions. In fact, I became so enthralled with these bacteriophage as a way of asking these fundamental questions that I ended up in a wet lab working with biologists and actually becoming one myself. And so I kind of call myself a biologist now. I've got the squiggly octopus head to represent the kind of trajectory that I obviously ended up down. And in fact, I am so much of a biologist, I feel like I'm still trying to convince people that that's the case, that I'm in the midst of setting up my own wet lab. At least I was until we completely shut it down in March. So what do I mean by the title of this talk? What was old is new again? And to illustrate this, let's think about kind of the trajectory of a field here. And so now a field is moving in kind of this theoretical space of concepts of different theories and norms that all kind of make up a discipline. So a discipline is this kind of organic-y blob that occupies some space in this kind of concept theory norm space. And maybe this is representing biology and it's moving through this space. And we don't really need to worry about why it moves or anything like that. But let's just consider this as kind of the evolution of the space that biology occupies as a discipline. And maybe you think about artificial life as this kind of fringe group of scientists that kind of were outside of the realms of biology and somewhere near the intersection of computer science and biology. But either way, the field of biology has progressed and has encompassed part of what I would say is kind of the bubble of artificial life. I'm not saying that artificial life is completely encompassed within biology. By no means do I think that's the case, and I think that's a good thing. But I think there are parts of artificial life's history that have become squarely within the boundary of biology. And this early research in artificial life, this place where people were studying questions that I now think squarely fall within biology, is a really interesting place to revisit. In part because I think there's answers to some biological questions that maybe were overlooked. And I think that's true in other fields as well. So I wanted to spend a little bit of time talking about some of these early spots. So one of the first that I think most of us know about is John von Neumann's work on self-replicating automata. And when he first conceived of these, he wasn't thinking about cellular automata. Initially, he was thinking of this as sort of a fluid or gas full of components, and it was assembled mechanically, sort of like arms reaching out and grabbing those bits and pieces to build up a new machine. And it wasn't until later that he kind of formalized this as a cellular automata. And I think one of the most fascinating things is that towards the end of his life, he was arguing that this is sort of the fundamental proof that machines, in theory, could evolve and increase in complexity. That there was nothing separating biological systems from these mechanical ones. And I think that's an early instance of some big questions that this field is still grappling with. I find it really interesting that Schrödinger is often credited with having the kind of premonition about the double-stranded nature of DNA by Watson and Crick. But at the same time, von Neumann was really kind of actively building the logical foundation of how information and a constructor could perform self-replication. And just for fun, I wanted to give a little history about some of the work that was being done at University of Michigan. Sorry, I'm going to get some flack for that, by some of these early figures in evolutionary computation and artificial life. So Arthur Burks was working with von Neumann to build ENIAC, and he was recruited to University of Michigan to start the Logic of Computers group. And out of that group came John Holland. And of course, many of us know John Holland as sort of one of the forebears of genetic algorithms, and also the advisor to some really well-known scientists, including, Joan Koza, genetic programming, Stephanie Forrest, who's well-known for artificial immune systems and building automatic programming and debugging tools with evolution. Melanie Mitchell, one of our keynotes that I'm just really geeked out, she's here, pictured here holding her new book, Artificial Intelligence, A Guide for Thinking Humans, but she wrote Complexity: A Guided Tour, which I'm sure is on most of our bookshelves. John Holland, here pictured with Arthur Burks at ENIAC, was really motivated by this idea of bottling up perpetual novelty of biological evolution and putting it into a computer. This is what a lot of John Holland's early work is inspired by. And another person, Chris Langton, who's working with John Holland at the University of Michigan, had another kind of self-replicating computational system that was actually implemented in a computer and was studied as a form of potential artificial life. And Chris is credited with organizing the first artificial life conference, catalyzing the field as a whole, and developing also a pretty compelling instance of artificial life with his Q loops here, that self-replicating automata. But in all these early instances of artificial life with John von Neumann's self-replicating automata, Chris Langton's loops, and even some later work by Steen Rasmussen in Core World, where computer programs written in Red code, which was the programming language used by Core Wars, a system sometimes called Core World, had this kind of brittleness. Evolution didn't really work. And this kind of raised this interesting question about where's the perpetual novelty, right? We bottled up evolution as far as we know, right? Evolution 101 is adaptive evolution occurs only if and only if there's reproduction, heritability of traits, and variation in individuals' reproductive success driven by those traits that vary. So what's missing, right? And I think Tom Ray was sort of the first person to clearly demonstrate what was missing, and I'll just let him explain it here.
So I thought about why it is that you can randomly alter the genetic program, but you can't randomly alter a computer program. What's the difference between these two languages that allows one to evolve but not the other? So I compared the two languages and came up with some ideas. One of the things I noticed is that whereas the genetic language consists of an alphabet of 20 letters corresponding to amino acids, the computer language has billions of letters in its alphabet. So whereas you could rearrange 20 letters and get a meaningful word, it's harder to rearrange a billion letters and get something meaningful. So the first thing I did was to reduce the number of letters in the machine language to 32, comparable to the 20 of the genetic language. Another idea that I borrowed from molecular biology is that when different molecules in a cell need to interact, they locate one another by having shapes that fit together. Whereas in a computer program, when different parts of the program need to interact with each other, they do so by specifying exactly where the other part is. But when things get moved around by mutation, those locations are no longer meaningful, so it doesn't work. So I borrowed this idea of shapes from molecular biology and arranged for my computer programs to locate the pieces they interact with by having something like a shape over there. So now when the pieces of the program get moved around, they can still find each other by recognizing the shapes.
And some of the people that I highlighted earlier also made appearances in this early video that I can send you a link to or send you a download for if you're interested. It's pretty fun. And around the same time, another group of scientists was working on an artificial life platform that I believe was ultimately influenced by Tierra, but kind of took its own stance on a few different design decisions. And this is, of course, what ended up being known as Avita. And this is a picture of some people that many of us know that are part of this community, at least 3 out of 4 of them on a regular basis. And for as cheesy as the video was by Tom Ray, I think there's a really important point that's made here. And I think it's so important that it's even been appreciated by biologists. So kind of highlighting that this is a journal specific to the organic evolution. So this is not a journal that maybe at least in the early 90s was amenable to these kind of computational instances of evolution. But in this quote in the abstract, I think does a good job of kind of summarizing what I think is a really fundamental contribution that Tom Ray and Charles Ofria and Chris Adami and others have provided to the field of evolutionary biology, which is that systems need this additional component in order to evolve. They need to be themselves evolvable. This isn't something that we would have figured out by just looking at biological systems, because they're obviously an example of an evolvable system because it did evolve. So this was really a unique contribution that Alife was able to make. And so much so that I'd say this kind of rewrote what the fundamental requirements are for evolution, right? In addition to having heritability and reproduction, you also had to have mutationally accessible variation. And that was an important distinction. And to me, this is really one of the biggest successes of Alife. And I don't think we give ourselves enough credit for it. What did the field of artificial life do once it figured out this fundamental rule about the way evolution works? Well, of course, it goes to look for some more. And following this thread, I think we're still waiting for artificial life to kind of wow us with the diversity and complexity of the living world, right? And, we found these really fundamental rules about the way evolution works, but really we're still kind of in this space where we've shown a flashlight into one spot of biology's potential for novelty. So the work that I became interested in is studying coevolution, how populations interact to drive more and more changes. So like in Alice in Wonderland, when Alice meets the Red Queen, she has to run as fast as she can to stay in the same spot because the world is constantly moving. Perhaps biological systems have the same problem, that everything around them is constantly evolving and changing the landscape that they exist in. So the only way to maintain your fitness in a world that's constantly changing is to also constantly change. This idea was first really explored by Danny Hillis, who's perhaps better known for his work with thinking machines and the connection machine, the sort of first stream processor, high performance computer with 65,000 CPUs. And what he did was look at a problem of sorting numbers and kind of in an abstract representation of which numbers do you compare? And he implemented parasites in this as basically a co-evolving test set. So which numbers are being tested against which sorting networks, which allowed the parasites to kind of identify weak spots in some of the evolved solutions and target those, and then drive the solutions to do better and better at that particular weak spot, and sort of over time improve the efficiency of the networks. And what he found was that coevolution was able to produce networks that were nearly as good as the best that had been found by humans. Now, there were other instances of coevolution being studied within the artificial life community, in particular with robots. So one of the famous examples are these predator-prey robots that were physical robots and competed in a real arena. And I think it's a really cool system, and it's really fun to watch. So I managed to take up this really old video of them. But you can see the predator robot kind of following with its eyes, at least, this prey robot, which can run faster than the predator but can't see as well. And so the predator can kind of sneak up, or at least the predator can try to sneak up on it. But one of the things that I really like about this particular setup is that these tethers that power the robots are also used to sort of reset the trials. So they get yanked at the end of the run, pull the robots back into the corner so that they can restart. If you have this arms race idea where you have to keep running in order to maintain your same level of fitness, then how do you test whether or not anything's getting better at all? And what Stefano and Dario did was they came up with this they used this approach of testing how well things did to previous generations. So take a predator from, let's say, generation 10 and compete it against prey from generation 5 and see how well it does, with the idea that if it's continuously getting better, even though you can't see it in the current generation, by looking in the past, you can kind of figure out how well it's doing. And these kind of time shifts where you're looking in the future and the past were a really powerful way of measuring progress in evolution. And so here, dark pixels represent places where the predator wins, and light pixels are places where the prey wins the particular interaction. And so it doesn't look like there's too much of a trend. If there was a perfect trend, you might picture this figure on the left where there's just a diagonal. But they didn't see that. And so one thing they tried was create a hall of fame where best solutions from previous generations were kind of stored in a virtual seed bank, and then they were added to the populations as evolution went on. When they did that, they find a more clear signature of kind of continuous change. But what I find extremely fascinating is that the same way of measuring coevolutionary adaptation was independently, as far as I know, discovered by evolutionary ecologists in the early 2000s. And this was at least 6 years after it was being used already in the artificial life community. This idea that coevolution might be one of the reasons why life is so complex was actually an idea that Darwin was kind of playing with. And so this is one of his handwritten notebooks, and it's really hard to read, but the enormous number of animals in the world depends on their varied structure and complexity. Hence, as forms become complicated, they open fresh means of adding to their complexity. But yet there's no necessary tendency in the simple animals to become complicated, although all perhaps will have done so from the new relations caused by the advancing complexity of others. So we get kind of a taste of maybe where Darwin was coming up with his tangled bank imagery, in this idea that the reason why life is so complicated and so interwoven is because life kind of threaded itself together by building it up. And so maybe coevolution, especially within this artificial life context, allows us to take that one spotlight and expand it a bit. I'm not claiming we're finding all of the complexity of the natural world, but we're certainly finding more by embracing the importance of these ecological interactions within the artificial life worlds. I'm going to shift now to talking a little bit about research that's going on in my group right now. I'm going to go through a real fast introduction to some of the older work on parasites in Avita, mostly to motivate some new work where I'm going to remove a lot of that complexity. So the computational work in my group is primarily done using Avita, which I showed you a picture of the creators of Avita both early and more recently. And in Avita, every individual in a population is a self-replicating computer program. They're composed of Turing-complete instructions that are executed on a virtual CPU, and the hardware that the organism has is capable of reading and writing instructions to different locations in memory and looping and sort of everything else you would need to write a self-replicating computer program. So here we have a host organism and showing its genome and its hardware and some of the logic functions that it's able to perform. And these logic functions are what enable the organism to interact with the environment. Within the neon rectangles, I've depicted some of the logic gate representations of these logic functions, just to show that there's differences in how complex these functions are. But in terms of what these functions do for the organism, they all do exactly the same thing. There's one type of resource in the world. Regardless of which task an organism performs, it tries to consume some of this resource. Now, there's also parasites in this world. The parasites are self-replicating computer programs that have to live within a host. They're effectively parasitic threads executing by stealing CPU cycles from their hosts. And instead of self-replicating into the world, they try and infect other host organisms. And so parasites are also performing logic functions, and the logic functions the parasites perform dictate which types of hosts they can infect. So here I've got pictured this pink parasite that's able to attach to the pink host receptor, or the green parasite that's able to attach to the green host receptor, or this kind of hybrid generalist parasite that's both green and pink, because it's performing both of those tasks, is able to infect through either of those. And there's also this kind of yellowy orange host task that is representing a resistant host. So hosts kind of do tasks that the parasite populations haven't evolved to target yet, and those hosts are effectively resistant. Here's a video of some parasites that get injected into a population that's been growing a little bit. And so this kind of gray circle is where the hosts currently occupy, and this bar is where we've injected the parasites. And in this case, parasites are going to be changing in color, and the color is representing the virulence of the parasite, how much of the CPU cycles they're stealing from the host. This isn't something that's evolving in most of the runs that I'll be talking about. It's just a really fun video that I wanted to share. So you can see the parasites sort of spread through the population, and these bright colors, the ones that kind of look like bright white, are highly virulent parasites that can quickly burn through the edge of the population and go extinct potentially. So some really fun work on the evolution of virulence, which hopefully I'll talk about during the Symbiosis Symposium tomorrow. But what's really useful about the system in Avita is that each of these logic functions has a well-characterized complexity to it. We know exactly the minimum amount of computation required to perform these different logic tasks. And that lets us do things like measure how much complexity changed when we have parasites or when we don't. And so if we look at how complexity evolves in the absence of parasites, we get this kind of drunkard's walk level of complexity. And it's a drunkard's walk because there's kind of a bounded lower level of complexity. So below some value, host organisms just can't live. And if we allow parasites in the system to co-evolve with their hosts, we see a much higher level of complexity evolve. And in fact, it looks like the evolved host line is kind of saturating while the co-evolved host maybe isn't. But let's kind of just ignore that for now. More than just this functional complexity, we also see a kind of complexity in the ecology evolve. And so here we have a video where parasite phenotypes are depicted on the top as red spheres, and host phenotypes are depicted on the bottom as blue spheres. And the width of the link connecting the two is the proportion of those parasites infecting that host. And we can see this really kind of large network evolve, and it's also quite dynamic. So it seems like it kind of stays stationary for a little while, and then a new parasite type or a new host type evolves, and it changes. So in this, let's watch this one more time. So at least in the beginning, you can see kind of a quick change while new phenotypes are evolving rapidly, and then you kind of get these complex networks that maybe are stable for a little while, but if we keep watching, we'll see a new parasite type evolve and then kind of shift the population into a new state. So these parasites and hosts are really creating an ecology that's quite dramatic and complex. And one thing that we found that was really shocking, and I mean this in the truest sense, like we weren't looking for evolvability, but we found it. What I mean by that in this case is that there are particular kinds of mutations that allowed hosts to go from doing one task to doing a different task without going through this kind of deleterious intermediate state where they're performing both of those tasks. You can imagine if there's parasites in the world, performing multiple tasks makes you susceptible to a larger array of parasites. And it was really only in the co-evolved case where we saw an enrichment of these kind of mutations. And that makes sense. This is kind of specific to this particular environment. And the way we found these is by looking at the mutational neighborhood, right? And so this kind of made me think this is a cool result. And usually we think about parasites being the evolvable partner. So we found evolvability in the hosts, but now let's go look for it with parasites. And in particular, this work is kind of motivated by a collaboration with some scientists in the Negev desert in Israel. And there's this interesting set of rodents that live there, and they live on different sand types. So well-compact sand, really loose sand, and kind of in-between sand. And these different rodents have overlapping ranges, and you can find instances of sort of all combinations of these. And so if you imagine a tick or some other biting insect that's carrying a pathogen, bites one of these mice or bites one of these rodents, transmits some pathogen to another rodent, and you can kind of imagine that depending on the different species of rodents that live in a particular area, this pathogen is constantly adapting to very different environments. And so this is kind of the situation where you'd expect evolvability to evolve. And so we want to test this using, well, both the system in the desert, the system of rodents, but also this computational system in Avita. But we're going to go even simpler than this kind of multiple populations of parasites representing different individual hosts. Let's just evolve a population in an environment that's changing. So we'll come up with two environments. So call it environment A, where the even tasks are rewarded and the odd ones are punished, or environment B, where the opposite's true. The even tasks are punished and the odd tasks are rewarded. And we'll flip back and forth between these multiple times. And importantly, we can do control environments where it's always environment A or environment B. And when we do that, we actually find something that's kind of interesting that I wasn't expecting, and that these two environments are different. One of them is harder than the other. And okay, maybe that isn't that surprising, but how dramatically different they are is surprising to me. And so we can do things where we're going to cycle between these environments, and we can cycle between these at different rates. So we could cycle between them at every 30 updates, 300, or 3,000 updates. And these correspond to roughly at every generation, every 10 generations, every 100 generations. And we can plot the fitness of population over time. Here, this is looking at the replication rate as opposed to sort of the merit, if for anyone that's familiar with Avita. But one of the things that we kind of notice right away is that in this environment where it's changing every 300 or 3,000 updates, these kind of higher ones, early on in evolution, they actually are adapting to this environment B faster than the ones that are only trying to adapt, that are only experiencing environment B. And that's cool, right? Half the time these organisms are being challenged in this environment, but the populations are adapting better to this environment when they have to go back and forth between two. This isn't a new phenomenon, but it's cool to see, right? So changing environments sped up adaptation. Cool. So let's dig into these populations. One way we can do that is by looking at what's happening through the line of descent. So looking at what happened from the very beginning of time in this evolutionary experiment to the very end, looking at only what happened along a dominant lineage's line of descent. And to do this, we're going to reference a particular environment, and this way we just have a point that we're measuring everything against. And we'll do this to environment A. And this is an arbitrary decision, but we can represent this environment as 1 0 1 0 1 0 to say that the first task is rewarded, the second task is punished, the third task is rewarded. And let's look at the different phenotypes, which tasks the organisms perform along the line of descent. So maybe one of these organisms is performing the first three tasks and the second to last task, and has this phenotype 1 1 1 0 1 0. That's one bit different from this kind of reference environment, and so we can measure this as the 1. You can imagine having another kind of 1 bit distance phenotype all the way to an organism that does the exact opposite set of tasks. And so maybe from environment B, this would be 6 bits different. And so we can characterize how far away the phenotypes are along the line of descent from this particular reference environment A. And so if we look at this in the case of changing the environments, cycling the environments every 30 updates, we see a distribution that's centered on 3. So most of the time, organisms seem to be kind of in the middle of the distance between A and B. If we start to look at longer times between cycling environments, we see this kind of spreading of the distribution where organisms now along the line of descent are falling either at 0 or at 6. So either all of the tasks appropriate for environment A or all of the tasks appropriate for environment B, which makes sense. They're spending time in those two environments. And as they spend even more time, we see those distributions spread even more towards those edges. So this makes sense. And it also means that organisms along the line of descent aren't really fixing one type. They're kind of going back and forth. So these are the kinds of dynamics that we'd hope to see if there was a chance that evolution could start to kind of adapt to having to switch back and forth between environments often, right? We want to see both of these environments represented on the line of descent. So now let's characterize the end result of evolution, this one dominant genotype, instead of looking down the kind of ancestry. And so we can picture this one dominant genotype as living somewhere in this kind of hypothetical fitness landscape, where the space is of different genotypes and perhaps the fitness values are these kind of ridges. And one way we can characterize some component of this fitness landscape is by kind of creating variation. So let's take this one genotype, create all possible single point mutations from it. And we can go even further and create all possible double mutations. And this gives us a sense of what types of phenotypes are directly accessible by mutation from this one genotype, this genotype that we ended up with. And if we look at the fitness landscape, you can think of this as sort of exploring the region directly around the dominant genotype. And so one thing we might want to know is how diverse are the set of phenotypes that are accessible by one or two mutations away from the kind of end genotype that evolution found. So looking at the constant environments, here's what we get. And when the environment's fluctuating, the diversity of available phenotypes, accessible phenotypes, is much higher. This is really cool. So fluctuating environments are selecting for genotypes, well, okay, let me be careful with how I say this. Fluctuating environments are pushing populations into a space where more phenotypes are accessible through mutations. That's cool. But I did have to deflate myself a little bit, because one thing I realized is that 30 update treatment, if we go back and think about the distribution of phenotypes along the line of descent, we saw that they were kind of centered around 3. So most organisms were kind of averaging the two environments potentially. And one way you can imagine averaging between these two environments is by performing all of the tasks, right? This is 3 away from both environments, and perhaps this is kind of the most adaptive phenotype. And it turned out that is typically what I was seeing in my runs was organisms evolving to perform all of the tasks. So I had to do another constant control where I reward all of the tasks. And in that case, the phenotypic distribution was higher, which you kind of expect. You have more tasks, there's more possible ways of breaking them, which leads to a more diverse set of possible mutations. So perhaps here we're seeing some effect of just having more tasks in this 30 update. But even for these 300 and 3000 update fluctuating environments, we're seeing an enrichment of phenotypic diversity. And in those environments, we're not seeing this kind of averaging anymore. But can we characterize more carefully the nature of this variation? And not just that it's producing diversity, but what's the structure of that diversity? And one way we can think about this is by looking at correlations between tasks that organisms perform. So here we're going to show a matrix of this particular quantity, which is the joint probability of seeing an organism perform task X and Y divided by the probability of those two occurring independently. And taking the log of that just kind of puts it at 0. So above 0 would be task X and Y are occurring more frequently than you'd expect together, given their independent probabilities. And a negative value would be that you're seeing task X and Y be performed less frequently together than you'd expect by their independent probabilities. And so this tells you something about how common tasks are co-occurring. And so here you can see most things are kind of roughly around 0. And the way you read this, it's symmetric, so you can just focus on one of the above diagonal or below diagonal. And if you say, let's look at, for instance, task 2 and task 4, here they're showing up together as being strongly negative, or not strongly, but relatively to everything else going on, negatively correlated, versus something like task 2 and 3, which is showing up as slightly positively correlated, perhaps. And remember that these matrix values are measuring the correlations between the tasks performed by all of the mutants generated from just a single dominant genotype from each evolving population. When we look at these constant environments, so places where only subsets of tasks are being rewarded, you can see this kind of checkerboard pattern emerge already, where there's some information, it seems, about the environment, the structure of the fluctuating environments. It's ending up encoded in the kind of mutational phenotype distribution from a dominant genotype, right? And this makes sense. In both cases, in both environments, const A and const B, even tasks are rewarded with even tasks and odd tasks are rewarded with odd tasks. So you don't expect to see these ones where there's an even and an odd. And in fact, we find a lower probability we find a deviation from the independent probability of seeing those two tasks in this phenotypic distribution. So organisms are less likely to perform these unexpected task combinations. But when we look at the changing environments that are fluctuating between which sets of tasks are rewarded or punished, we see even more structure in the distribution of phenotypes being produced by these all possible single double point mutations from a single genotype. So it seems like there's some kind of information encoded about the structure of the environments that are being rewarded that's ending up in the genotype phenotype map of these organisms. I think that's really fascinating. So another way we can look at this data a little more quantitatively is by just looking at the deviations from independence that we might see between pairs of tasks that are either expected to show up together or unexpected to show up together. And because I've kind of grouped them as the unexpected tasks, I flipped the sign so that they're all the higher positive values mean more information about the structure of the environment. So through the process of adaptation, evolution has moved these genotypes into a space in the fitness landscape where the distribution of fitness effects matches the structure of the environments that they're experiencing. And that information is showing up in the genotype phenotype map, in the distribution of phenotypes that are accessible by single and double point mutations on just a single genotype, that's kind of the dominant genotype at the end of the run. Monica Acosta is a postdoc that just joined my group recently. And when we were sitting down and talking about this data that I just presented, she became really curious about how different forms of evolvability might interact during evolution. And so partly that's because one of the first mechanisms that you usually think about evolvability in terms of is mutation rates. So organisms with higher mutation rates can explore a larger region of that fitness landscape and expose that variation to evolution to kind of adapt even better. And so how does that kind of evolvability interact with this kind where we're thinking more about the genotype phenotype map? And so this is a question that Monica became pretty interested in and is working on right now. So to get a baseline of how mutation rates influence adaptation in these kind of fluctuating environments, Monica ran the same kind of variable environment experiments that we've been doing here, but with a range of mutation rates. And as you might expect, organisms do best at some sort of intermediate mutation rate that's not too high. So you can see when the mutation rate's too high, everything kind of dies out, or at least doesn't adapt, which makes sense. When the mutation rate's too low, things are adapting, but they're taking a really long time. And there's some sort of intermediate value that seems to do well. And in this case, it's about 1 in 1,000 here, this 1e minus 3. So we can maybe call this an optimal mutation rate, although I'm going to use air quotes, even though you can't see them. And immediately we found something cool. So even if this is an expected result, it was cool to see that genotypes from higher mutation rate environments were more robust. They had a higher proportion of genotypes in their mutational neighborhood that were viable. And this sort of makes sense. If you're evolving in a world where there's just constant threat of mutation, you want to be robust to that mutation. And we know this is an effect, and partly we know this is true because of experiments done with Evita on survival of the flattest. But it was still cool to see sort of in our hands, right? And in a sense, you can think about this as kind of similar information. This is information about the environment, high mutation rates, low mutation rates, that's getting encoded somehow into the genotype phenotype map. But one of the things we know from also previous work with Evita as well as other systems is that evolution tends to select for lower and lower mutation rates. We don't tend to see evolution picking kind of the intermediate mutation rate or the high mutation rates, even when we can show that in that environment, a higher mutation rate leads to better adaptation. We didn't have high hopes, but Monica was interested in seeing what would happen in these environments if we let evolution tune the mutation rate. It's an environment where we already know evolution has favored evolvability. And to my surprise, evolution actually did lead to higher mutation rates, or at least not as low mutation rates in these fluctuating environments. And I drew a line here at this 1 in 1,000 rate, which we identified as sort of the optimal, again with air quotes, mutation rate for some of these organisms. And it seems like in these fluctuating environments, we actually maintain appreciable mutation rates, even perhaps close to that optimal mutation rate. And this was really surprising to me. Now, we're still working on figuring out exactly how to tease apart these two different kinds of evolvability. So how does an evolving mutation rate affect how you incorporate information into the genotype phenotype map? These are things that are still a work in progress, but hopefully you'll get to read about them soon. And so I just wanted to wrap up by saying, we're still after this perpetual novelty of biology. This is kind of, I think, still one of the guiding principles of at least my work within artificial life, as well as a growing community of us. But I think we also have to acknowledge that we're getting closer. And so with that, I'd just like to, again, thank the organizers, thank all of you, and happy to take any questions.